I need help.
Here's the problem. I'm trying to figure out how I should eliminate a specific gene in a eukaryotic cell for a research project to see what the phenotype will be if the DNA lacked the protein. I have two options. I could use RNA interference or CRISPR. And to help me, I'm going to make a Venn diagram. So let's get to it.
Here's my Venn diagram, so let's get started.
One difference is that RNAi targets RNA and CRISPR targets DNA. RNA interference uses a dsRNA produced in a lab and is cleaved by a dicer, an enzyme that turns the dsRNA into either siRNA or miRNA, which is around 21 nucleotides long. From there, the modified RNA is bound to a silencer called RNA-induced silencing complex, or RISC. This inhibits the translation of miRNA or siRNA. CRISPR uses a guide mRNA to create in a lab. The guide mRNA binds to the Cas9 enzyme to recognize and cut the desired target sequence or gene of the DNA. DNA then repairs the break naturally by copying template DNA and inserting it into the break. The template DNA can be supplied either by the cell or researchers, but since we are only looking for the effects of inhibiting one gene, we will let the cell fix the break. I just mentioned a few more differences, so let's add them. RNAi uses dsRNA. Here is the differences that I just said while explaining the two methods. Another difference is that RNAi's effects are only temporary. Since it's only being silenced, the genes are still present, and the phenotype is only being masked. Since CRISPR deletes the sequence, the results are permanent. RNAi is also a much more simpler and quicker process than CRISPR, so I will have my results sooner, but time isn't an issue for me. Another difference is the amount of errors that can occur during gene editing. The complete removal of the gene or genes during CRISPR results in less off-target effects. Some may still occur, but is more likely to occur in RNAi. In RNAi, the silence of the gene can silence other proteins and genes that affect the phenotype. I only want to see what happens when my desired gene is removed and no other changes occur. CRISPR is also newer. It was invented in the 2000s, while RNA interference was invented in the 1980s. However, both methods use unmodified RNA to inhibit gene expression. Another similarity is that both methods can result in false positives and off-target effects. CRISPR can have off-target effects if a mutation occurs when the cut from Cas9 is being fixed by the cell. The cell can either glue the ends of the broken pieces together or insert a template. If the cell decides to stick the ends together, mismatching may occur and result in a mutation. So I decided I'm going to use the CRISPR method. And that's just because that it is more precise, even though it takes longer. But I don't want any off-target effects or additional mutations. I just want to see what happens to the phenotype when I remove that one gene. So yeah, so thank you guys so much for the help.